today I'm excited to announce a QA talk. Katie did her BS at Sharpie Mud, after which she was a technician with Kathy McFadden, who came to Davis as a postdoc, so following a little lineage there. After which we were lucky to attract her here. She's brought her technical skills, positive energy, and evolutionary thinking to our lab and to Pop Bio more generally. It's really been a pleasure to have her so far. She'll be talking about what I think is a pretty cool integrative dissertation looking at structural variation in local adaptation.
Thank you, everybody, for coming today. Here is the title slide for my talk, but just to take one step back, give you a little bit of an outline. I am going to talk about my dissertation research today and talk about proposed research, share a couple of preliminary results, and have some time for questions. But I do want to save about 10 or 12 minutes at the end to talk about another project that I showcased earlier this month, called Exactive Potential, which was an eco-trash fashion show. And I'll be giving some artist statements, showing you a little behind the scenes film, and there is some show and tell. So I'm going to keep the research part a little bit shorter and sweeter today, hopefully to leave time for that. But back into my dissertation research, as Rachel mentioned, my research is focused on the eco-evolutionary genomics of a large chromosomal inversion in California eelgrass populations. So the kind of central evolutionary process to my dissertation is local adaptation, in which a population, individuals in a population adapt to have highest fitness in their local environment. The figure on the screen is the kind of canonical representation of this process, in which population A has a higher mean fitness in habitat A, and the same for population B. This type of relationship can be seen most directly through reciprocal transplant experiments, in which each population is placed into each habitat and their fitness traits are measured. And this type of relationship was first identified in natural systems over a century ago. But only in the last couple decades have researchers been able to kind of uncover the genetic basis of this process. Related to that process of uncovering the genetic basis is a question of how repeatable the process is. When I think about parallel local adaptation, I think about when closely related taxa respond in the same or similar ways to similar environments. So the model here, you can see there's like two recently diverged taxa, and then they're following similar evolutionary trajectories. This is something that has been established in many natural systems. One of my favorite case systems is a dune and headland ecotype of an Australian wildflower. So you can see that the dune ecotype and the headland ecotype are they're distributed across the entire east coast of Australia, where you'll see a dune and a headland ecotype very near to each other. But when you plot the relatedness of these individuals on a tree, you see that that ecotype differentiation has evolved multiple times. So in this system, we can think that these wildflowers have adapted to these two different environments in parallel. And usually the kind of underlying data to these questions about the genetic basis of local adaptation and parallel evolution have been based on SNPs, or single nucleotide polymorphisms. So these are single base pair mutational differences between two sequences of DNA. But recent advances in sequencing technology, as well as bioinformatics tools, have allowed us to capture structural variants, which I'll sometimes refer to as SVs, which are these larger scale changes in the genome. There's a bit of a range of cutoffs that people like to use to define SVs. I've seen numbers all the way down from like a 50 base pair variant to some people want it to be 50,000 kilobase or 50,000 bases or longer. So know that there's some wishy-washiness there of what constitutes an SV. And there's also a lot of different types of structural variants. from, we have inversions, translocations, fusions or fissions. CNVs are copy number variants, and also we can think of polyploidy as this model. But for my dissertation, I'm focusing mostly on one of these, which are the inversions. So chromosomal inversions are mutations that occur when a portion of the chromosome kind of switches its orientation and reattaches into the genome. So we can see the inverted region is in orange in this diagram. And these two chromosomes have the same genes, but this one region, the orientation of those genes is reversed. When an individual is heterozygous for an inversion, crossing over is really reduced in that inverted region, and that allows for co-adapted alleles to be transmitted reliably. What that means in the case of this figure is that you could kind of generalize that in heterozygous individuals, they would pass down that entire orange block as one large locus or one set of genes. And so very large chunks of the genome can kind of be inherited as one unit when you have this system. And this recombination suppression has been shown to have many interesting eco-evolutionary implications across diverse taxa, anything from impacting mating systems, behaviors, and phenotype-genotype associations. The system that I'm interested in studying these processes and these types of variances in is Zoster murrayna. You can see this beautiful eelgrass bed picture here. Zoster murrayna is also called eelgrass and is a marine foundation species that supports a lot of invertebrate and fish populations. And it's also very important for carbon sequestration and nutrient cycle and coastline protection. Because it's so ecologically important, and it's also been experiencing declines across its range. It's the current focus of many different scientific, conservation, and restoration efforts. You might see this picture and think, wow, that's so beautiful. I want to go snorkel in an eelgrass bed, but I will admit that the reality is most of my field sites look a little bit more like that. But if you're still enticed, I will bring you out anytime. Zoster murrayna has a very large range across the Northern Hemisphere. So it grows intertidally and subtidally along coastlines across most of the Northern Hemisphere, which means that it spans very variable environments. The temperatures that it experiences ranges all the way from 0 to 40 degrees Celsius, and it has similarly drastic differences in salinity, light availability, and its nutrient environments. A little bit about the biology. So Zoster murrayna is a marine angiosperm. It's not kelp, a common misconception, which means that it produces flowering plants. I will admit they're not the most beautiful flowers that you've seen on a plant. Up in the top right corner here is an image of the female flowers. You might not recognize it as a flower, but now you can. So it does experience sexual reproduction, and it also grows asexually by producing multiple shoots off of one below ground rhizome system. So in this drawing, you can see there's the below ground. The kind of orange part is a rhizome with roots growing out of that. And then there are multiple shoots with multiple leaves that grow there. The kind of final detail that I'll orient you to in these photos is you might notice that there are some brown blotches on this eelgrass drawing. And those represent disease lesions caused by eelgrass wasting disease, which is a disease caused by a protist and is very prevalent along the California coast. We affectionately refer to it as labby because of the protist that causes it. So that's a lot about Zostra. And now I'll talk a little bit about how that relates to these processes of local and parallel adaptation. So eelgrass, there's already studies that demonstrate that it shows very fine scale local adaptation. One of them out of Tomales Bay from Katie Dubois, a former StackLab member, did a reciprocal transplant experiment where plants were taken from 3 sites and outplanted back into those same 3 sites. If you look at the plots here, you can see that the survival for each population was highest at its home site. So that home site advantage is kind of the canonical relationship that we'd expect, and this kind of showed in a very direct way that eelgrass does experience local adaptation even at these very fine spatial scales. And eelgrass also adapts to temperature in parallel, and that was shown in 2 bays. So this figure here shows Tomales Bay kind of in the south and Bodega Bay up north. Importantly, the northern section of Tomales Bay, closer to the water, kind of mirrors a similar environmental gradient as the smaller Bodega Bay does from its mouth to its head of the bay. And when looking for outlier SNPs associated with these temperature gradients across the 2 bays, over 5,000 SNPs were shown to be shared between Tomales Bay and Bodega Bay. You might think that that is really good support, that there's very high parallelism between these 2 bays, but there's something else going on with this story. These 5,000 shared SNPs were actually found in just 1 linked region of the genome. And when you pull that away, you see that there's actually very little signs of parallelism or shared SNPs across the rest of the genome. So what's going on with this linked section? Really, the story here isn't that it adapts in parallel, but that there is a putative inversion that co-varies with temperature gradients in these 2 bays. So this is a Manhattan plot of the same outlier analyses that were shown as Venn diagrams earlier. And you might notice circled in red, there's this region on chromosome 1 where a whole block of DNA is all coming out as very strong outliers associated with temperature. And there's very high linkage throughout that whole region. If you break that region out and run a principal component analysis just on those SNPs, you end up seeing something that is a very inversion-like PCA, where we have these 3 very distinct clusters on that main axis of variation. So on the right and the left, you'll have the homozygote genotypes for the inversion, and then the heterozygotes all in the middle. So that's kind of where I came into this system of joining the Bay Lab, and there was an inversion that had been identified, but not much beyond that had been done. And that's where my dissertation kind of starts. So I have my 3 chapters, and as usual, I'm going to start with the first one, which is the genomic characterization and evolutionary history of the inversion. To get a good kind of groundwork for the rest of my dissertation, I needed to validate that this linkage block was really an inversion. It would be useful to know kind of the breakpoints where it starts and stops. There's a lot of descriptive work that I am working on. But for today's talk, I'm just going to share 2 little tidbits of results. This chapter has turned into a very collaborative project that's kind of intertwined with an effort to get a new Zostra reference genome out. So there's a lot of cool things here that I hope to share later, but I'm just going to share a couple things that I think are relevant. The first one is that our inversion is an inversion. So using the new references assembly for Zostra Marina, this plot essentially lays out 1 haplotype, so 1 assembled haplotype of chromosome 1 on the x-axis and the other on the y-axis. When you have a green kind of diagonal line straight through it, that shows that there's high sequence similarity and those areas are collinear. So most of the genome looks like that. You might notice there's this kind of yellow fuzzy area in the middle. We think that's because of the centromere region where sequencing and assembly is a little bit disrupted due to these repeat sequences. But what I really want you to look at is up in the top right corner. We have this region where the sequence similarity is still very high, but the whole region is just like flipped 90 degrees, which means they're like antilinear rather than collinear. And this is kind of the the main analysis that we wanted to run to show that this is not just a linked region, we do have an inversion here. So that was great. Some other work that I've done in this chapter, slide is weird, is trying to figure out some more about the biogeography of inversion genotypes. So before I joined, some undergrads had worked on genotyping the inversion across some California bays, and I came and added to their sample sizes and their sampling locations, and ended up with this kind of map. Some of the interesting things that I want to point out to you is that the zone of heterozygosity is not some neat mixing zone. We have Tomales Bay, Bodega Bay, and Drexel Cerro all located near each other, and then Morro Bay is like way further south, and there's these monomorphic bays in the middle. We've been calling this dark blue orientation that exists both in Monterey and San Francisco Bay, but also Humboldt and all the way north up to Washington, our northern orientation. So I'll be saying northern type a lot this talk. And then one that is only found monomorphically in San Diego as our southern orientation. Another little interesting tidbit here is that in Tomales Bay, we're seeing a huge excess of heterozygotes. So this is based on some genotyping we did that found 68% heterozygosity. In samples we've done in individual areas there, we found up to 80%. So this like huge deviation from what we'd expect under Hardy-Weinberg is a good indication that there might be some sort of selection happening upon this locus. So as I mentioned, that's all for chapter 1, keeping it short with just a couple results. But next up, I'm going to talk to you about the functional impacts of the inversion on gene expression and phenotypes across temperature. And some background, oh, sorry, that transition got mixed up. As happens in this system, outlier analyses are often the way that inversions are kind of detected in the first place. People go looking for outlier SNPs, they find a large linkage block, and they find a putative chromosomal inversion. That also makes it really hard to understand what specific genes or variants in the inversion might be contributing to functional differences or impact their fitness, because we can't disentangle those SNPs from one another. So in this case, our chromosomal inversion, everything comes out as being associated with temperature, even if some of those SNPs might be neutral. But one thing that we can do is use gene expression analyses to help disentangle that. Look for genes that are actually differentially expressed across genotypes, rather than just looking for these associations. I approached this by running a mesocosm experiment at BML, and I will be walking you through what that process was like. So almost 2 years ago, in June 2023, I collected 400 plants from one site in Tomales Bay, and I planted them in these outdoor tanks at BML. As you can see, we had a really huge field team out there, which was really great for one of my first big field pushes. So it was lovely to have so many people and waiters. And I also want to give a huge shout out to Carolina, who really showed me the ropes at BML and helped me with this experiment. She has like this huge sediment tank behind her, and we like hand scooped so much muddy sediment from the field that they had to use a forklift to get it out of the truck. So that was a feat. While the plants were sitting in the tank, I came back to Davis, and I genotyped all of them using a restriction fragment length polymorphism assay. So I went in the lab, amplified a certain region of the chromosomal inversion that had a fixed SNP between genotypes. That means that if you apply an enzyme to that PCR band, some of the genotypes get cut into multiple products, while some do not. So in this representative gel image, you can see that the N, or the northern genotype types, have this one large band, whereas the southern genotypes get cut into 2 smaller products. And then our heterozygotes that have both haplotypes show all 3 bands, because they have a mix of both of those. I also, during this month that I genotyped 400 plants while they were in experiment, took the core exam. So that was fun. With all of my plant genotypes, I actually put plants into treatment. So I had 2 tanks at a control or ambient temperature and 2 tanks that were heated. And well, these are the temperature profiles for those tanks where the blues are the colders and the colder tanks and reds are the hotter tanks. And while they were in experiment, I measured their growth, leaf length and width, as well as disease severity. So I had kind of a moderate subset of traits to look at alongside this data. Upon breakdown, I was flash freezing tissue for each plant, which will eventually be used for RNA sequencing. So the 4 tissue types I chose were healthy young leaf tissue, a diseased leaf tissue. So we found the youngest disease lesion of the protists that I mentioned earlier. The undiseased leaf tissue is an age-matched kind of reference for that diseased leaf tissue. And then finally root tissue by grabbing the first full root bundle for each one. Next, I ran into a little bit of hiccup because at the same time that I was about to start my RNA extractions, Brooke in my lab was getting some sequencing data back from the same site that I collected my plants for using a very similar sampling mechanism and found that her data had a bit more clones than I was comfortable having in my RNA set. So I put a hold on the RNA extractions and I used reduced representation sequencing to detect clones in my data set so I can move forward with a clone-free data set and not lose any of my sample size due to clone mates. So I went through all of that just to find 0 clones. It's really a good way to spend several months and several $1,000s on sequencing, but it was also great because I didn't have to reduce my sample size as much and I was able to go forward with my RNA extractions and sequencing. I submitted my 188 samples to the Joint Genome Institute in January, and I got my data back last week. So Wednesday, I got my sequencing data, and JGI actually went ahead and analyzed all of my data together as just one group. And so I have one figure to show you, which is the PCA codes for differentially expressed genes. And what we can see here is that at a first pass, everything really separates by tissue type. So on the right, we have the root, and on the left, we have the 3 different types of leaf tissue that I had. So you can see that PC1 kind of separates root and leaf, and then PC2 separates the diseased, the healthy, and the undiseased plants. So that was reassuring, but I do need to dig in and run these analyses actually separately and separate the groups the way that I want so that I can see hopefully the combined effects of both treatment and genotype on this expression data. Some of the other questions that I'm hoping to answer with these data are, does the inversion contribute to cis or trans regulatory effects? So if the inversion has trans regulatory effects, we'd expect that differentially expressed genes are distributed across the whole genome. So this means that even though the variation we're interested in is in this inversion locus, the effect it has cascades across the entire genome. The opposite of that we might see are these cis regulatory effects where differentially expressed genes are concentrated in the inverted region. In other natural systems, finding these cis regulatory effects in specific tissue types or life stages has helped uncover the function of the inversion because you can tie it to a more specific place in the genome. Another question I'm interested in is, does allele-specific expression contribute to phenotypic differences? So in inversion heterozygotes, they have both haplotypes of the inversion, and expression levels are not necessarily equal across genes from each haplotype. In a system of seaweed flies that has an alpha and a beta haplotype of the inversion, across many different genes, they saw this uneven transcription. And I'm interested in seeing if that's happening in my system. I'm mostly interested in it as a potential adaptive mechanism. As I mentioned before, we had this kind of huge heterozygote excess, and you could think about being able to express different genes based on which haplotype is more favorable as a way for heterozygotes to actually confer the advantage that we might be seeing based on our genotype frequencies. And that's the end of chapter 2. Moving on to chapter 3, I want to look at the impact of the inversion on parallel evolution across multiple California bays. So my third chapter, you can tell how happy I am in the field. I like to think that this chapter was like brought to you by swimming. I had previously only done eelgrass research in waders during really good low tides, but I wanted to go all the way up and down the state of California in 1 month and collect every day. And so I realized that I could do that if I just got wet. I was very cold there, and I did eventually get a wetsuit, which helped a lot. But that was very fun. Also fun because I not only convinced myself to swim, but I convinced Deva to swim with me. So all of the field work that I'm presenting was done in collaboration with Deva, who also took replicate samples for microbial analysis. And Kendall didn't jump on the eelgrass train, but she has the wetsuit and swim skills if she ever decides to. So going back to kind of the intro of my talk, I want to remind you about what we do know about parallel evolution in Zostra, which is that it seems that all of it is kind of congregated in this inversion region. If you look across the whole genome, or if you exclude the inversion region, we weren't seeing high signs of parallelism between just these 2 bays, but it seems that the inversion is kind of shifting in genotype frequencies similarly in these 2 bays. This relates to some background theory on parallel evolution and inversion. So case studies, theory, and simulations have all indicated that inversion polymorphisms might increase the probability of parallel phenotypic evolution. And to kind of generalize the logic behind this, if you think of the inversion as like one large effect locus, then changes in that one locus might shift some population mean to match their local environment the same amount just by changing this one genotype frequency than you might expect if you see a polygenic response of many small effect loci across the whole genome. So if you could just change like one locus, that might be a relatively easier thing to do than changing, hundreds at once. So my sampling approach was to look at bays across California. I wanted bays that had similar environmental gradients, which generally meant I want bays that were large enough to have marked differences between the mouth of the bay, where they're getting a lot of ocean input. There might be colder temperature, more wave action, higher salinity, versus the back of the bay, where things tend to get very warm, very mucky, more turbid. And so I chose 6 bays, ranging from all the way north in Humboldt and all the way down to San Diego. Note that I have all of the inversion genotypes present here. So we have Arcata Bay and San Francisco Bay, which are monomorphic for the northern inversion type. San Diego is our one southern bay. And then I have 3 polymorphic bays in Tamales, Drake Cestero, and Morro Bay. So in each of these bays, I sampled 5 locations spanning from the mouth and the head of the bay that have, as I mentioned, these similar environmental gradients. And I collected 20 tissue samples for each. So each bay has 100 plants for whole genome sequencing. In my bays that are polymorphic, I got some more samples just to genotype the inversion so that we can have a more robust sample looking at those changing genotype frequencies at that one locus. And then I also collected some morphometric data and deployed temperature loggers for 1 year. This means that this spring I'm doing 1,020 DNA extractions for this chapter. So I have been chronically in the lab. Some of the questions that I'm hoping to answer with this data set are inversion genotype frequencies correlated with environmental variables? So what we might expect here is that at the cold mouth of the bay, we have more of these northern types. They come from northern latitudes where they're exposed to colder temperatures in general. So that same latitudinal climb we kind of see on a large scale across the whole West Coast might be happening at a smaller scale within these bays. And we might see more of the southern alleles in the back of the bay, the northern alleles near the mouth of the bay. I'm also interested in if the inversion region underlies local adaptation in monomorphic bays. So in bays that don't have any polymorphism, they only have one of the inversion type, is that region of the genome still special and still kind of implicated in a local adaptation? This trend has been shown in southern sunflowers, where some populations that had a inversion that was locally adapted to an environmental gradient. If you look at just that region of the genome, other populations that were lacking that inversion still had a higher rate of outlier SNPs or SNPs associated with local adaptation in that genomic region. That might indicate why this inversion has stuck around, if there's just something about that region, some gene in there that is kind of really important to this process. Another question I'm interested in looking at is what biological scale will we see evidence of parallelism? So when you're looking for parallelism, you can think on the really small scale and ask, do these two populations adapt using the exact same like SNPs, the exact same variants? You could also scale out to many different levels and see, are there different SNPs but within the same gene leading to similar outcomes? You can scale back even from that and look at the molecular pathway and keep going into like molecular networks. And you can also look at phenotypes. As a reminder, excluding this chromosome 1 region, the SNP and gene level has been looked at for 2 California bays, and it was shown that there was very little overlap in those, just moderately more at the gene level than the SNP level. So for my work, I might expect that those changes can be seen kind of on higher levels of organization, more at the molecular pathway than at these fine scale biological scales. So with that, I'm going to get into my acknowledgements. I want to thank my advisor, Rachel Bay, as well as my QE committee. I have patched together many different funding sources to support all of this fieldwork and sequencing, and across the Bay Lab, also especially people in Anya Brown Lab and Jay Stakowitz's lab have been hugely helpful in all of my experiment and fieldwork, and the Pop Bio grad group. Yeah, thanks everyone.
So we're doing science questions.
Yes,
Okay.
I'll take a couple of science questions and then I'll save about 10 minutes to talk about art. Yeah.
That was a really awesome talk and it's super interesting. I kind of have a two-parter question that I was hoping. I don't want to take up too much time, but first part of the question is, do you think there was a single inversion that spread through the populations, or is it repeatedly getting inverted in that spot?
That's a good question. Some of the work in chapter 1 that I didn't show you kind of goes into that. We know some about the colonization history of Zostera. So its native range is in Japan, and there are hypothesized, or there's been shown to be kind of 2 introductions, one that's much older to the US West Coast. And so fitting in where the inversion arose with that story kind of contributes to a leading hypothesis that after the first introduction to California, the inversion actually occurred and ended up fixing in San Diego. And then this mixing zone that we have is kind of these San Diego inverted types coming up the coast at the same time as the second introduction from Japan reached Northern California and even north of that and coming down the coast. So I think just because of the, I think just like probabilistically, it probably hasn't happened twice. I think it's just one.
Awesome. And then the follow-up to that, if you don't mind, is given how patchy some bays, had fixed, some didn't, some were admixed, how much of that do you think is due to like microhabitat, like this is just a colder bay, versus the current going up California has eddies or something that reduces the chance of dispersion into reflective bays?
Yeah, that's a great question. I think San Francisco Bay is one that like really pops out as like, why is there not heterozygosity there? Why is this one bay that's kind of surrounded by other polymorphic areas not polymorphic? I don't know if I have a great like guess as to why. There's some things about San Francisco Bay that make it unique. It's very turbid compared to some of these other bays that are slightly more pristine. But one of the things that I do want to do to kind of get at that question is see how much, since the San Diego plants are more distinct from the other populations, we can look at the rest of the genome and see, are San Diego alleles generally making it up to San Francisco? And we believe maybe the inversion did too, but it didn't stick around. Or is San Francisco just like not even like having the migration, having those plants enter the bay in the first place? And that should get at your question.
Thank you. Yeah, that was awesome.
Super cool, really fun research. I'm excited to see what you find with the rest. It's very similar to Peter's question. I know there's a fair amount of restoration that happens with zoster, and I'm wondering how much movement for restoration there has been, and how that, like how well it's recorded so you know when it's happened and where it is and how you deal with that.
Yeah, that's a great question. I know that will come into play for my Drake Cistero plants. So Drake Cistero, maybe about half of the eelgrass in that bay, it has been recently like replanted out there. And that's one place where they did keep pretty good records of where the plots are. So I think I'll be able to get at that a little bit. Yeah, restoring and transplanting eelgrass is definitely something that happens and will impact my data. And I probably need to think a little bit more about that, but I am especially aware of it in Drake Cistero. I think another kind of difficulty is not necessarily knowing, even if we know where they planted plants for restoration, not like knowing where they gathered those plants from. So that will be something I'll need to keep in mind.
Super fun. The excess of heterozygotes is kind of bizarre.
Yeah.
Are you thinking that that has to do with, I guess, differential survival, or could there be something about this locus that's involved in like mating patterns or sort of mating incompatibilities?
Yeah. I don't know if my dissertation will address if there's like differences in mating compatibilities. One thing that I would like to do to kind of look at kind of how the selection, or if there's like survival that's playing out, is one idea I had is going through and genotyping seeds. So we should be able to genotype the inversion both in a parent plant and for its seeds, as well as know like the population genotypes, and see if at least the seeds are kind of segregating as we'd expect. If there's something happening at that very early life stage, we might be able to detect that. So that's like one of the ideas I have to get around that. But I would love to hear if you have other ones. We should chat.
That sounds great.
Yeah. Okay. I have time for one more question. Yeah.
Well, there was a question online about the outlier analysis that you mentioned in the first part of your talk, and how sensitive it is to differences in sample size between groups.
That's a good question. The sample size for that study was fairly large. So that was 288 plants whole genome sequencing. So I don't think in that study that sample size should have too large of an impact on that. I am, as I mentioned, I collected extra plants in my study on parallel evolution in just the polymorphic bays. so that when we're looking at just one locus, we do have like a really high sample size if we want to see changing inversion genotypes just in one place. So make sure you have a lot of them. Okay, well, I would love to talk to y'all about art for a little bit. So as I mentioned earlier, I will now go into talking about exaptive potential. So there are going to be some objects passed around. You're welcome to interact with them, touch them. They're a little bit fragile, but you'll hear more about what they are as I talk. On April 5th, I collaborated with Mae Blundell and Tracy Hayes to host this event, Artistic Visions of Coastal Renewal, at the Bodega Marine Lab as part of our Belinsky Fellowship fellowships, which combined art and science. My project was called Exaptive Potential, and it was an eco-trash fashion show by N Girls Collective, which is my art collective with myself, Anna Getter, Erica Goodwin, and Sitoe Sham. We have been making art for and with each other for almost a decade, so coming together to do this project on a larger scale was a really amazing experience. I'm going to read out the kind of artist statement that we read before a performance, and then show you a video, a video of the actual event day with some behind the scenes photograph. So you can feel like you're getting the full experience of when I spoke about this on event day. Our runway show, Exaptive Potential, is based on the evolutionary concept of an exaptation, which is when a trait evolved for one specific function takes on a new role. An example is that bird feathers were first evolved to provide warmth, but their lightweight structure ended up being co-opted to allow birds to fly. In a rapidly changing world, we hope that organisms can adapt to new environments, and we dream that they may reinvent their survival skills to exapt as well. We as humans can create exaptive potential by finding new functions for the things that exist around us. In this show, we did that at the Bodega Marine Laboratory and the surrounding natural environments. We'll share just a few of the exaptive potentials we uncovered, so you can look out for them when you watch the show. The first is agar, which is a gelatinous substance derived from red algae. Red algae gets its strength and elasticity to be able to survive in rough intertidal environments from this substance, and then scientists have already exapted it to use in the lab to grow and cultivate microorganisms or run electrophoresis. In this project, we find exaptive potential in agar by using it to create bioplastic material and give structure to knit clothing pieces. Also, garlic bags. You might recognize this mesh from garlics that you get at the grocery store. When we went to Bodega Marine Lab with my collaborators, Joe from the art department gave us a tour of his personal materials collection. He found this to be useful in aquaria because it could be used both kind of as a rope and be very strong, but also it was a net material, so it could have things flow through it or even hold things. And we found some fun ways to use that in our collection. And then finally, bioballs. Bioballs are used in aquaria to provide surface area that support growth of good bacteria and promote healthy nitrogen cycling. There were bats and bats of bioballs out in the dumpster by BML, and we thought that we could make them beautiful. There's a couple different forms, so you can see that they were sometimes made into textiles themselves, or they were used to print onto fabric. So next up, I have a video of the actual performance, and I'm just gonna talk on top of it. So you'll notice the models are very familiar faces. They're all grad students from CPB. So first we have Shreya. She's wearing a gown that was cyanotyped with bioballs, which was a very beautiful process. Up next, we have Kindle, who is wearing a net skirt and this top. And I brought my collaborators out in waders, which was very special. And we had a lot of fun working with net, although sewing it was quite tedious. Up next, we have Natasha, who is wearing the agar knit shirt that has been passed around, as well as a cyanotype skirt. So we actually knit a shirt and then set it onto a mannequin using agar, So just like you would make a gel in lab, except for painting it onto a mannequin. That one was passed around. Up next, we have Serena wearing a shirt made out of bio balls over a dress. And so figuring out how to turn these small beads into a flexible structure was a fun challenge for one of my collaborators. There was also just this fun smocking detail that you couldn't see on the dress, so I wanted to pull that out. Next up is Kenzie, who's wearing a top made of shells that were actually the shells of organisms from experiments at BML. So you'll notice they have the classic little nail polish tagging on them. This was another material that was challenging but fun to figure out how to turn it into more of a textile by drilling many tiny holes and looping them all together. Up next, we have Deva, who's wearing a net dress with a cyanotype gown. We found different ways to kind of overlay the net to give it different structures that you could see through. So it has this diamond pattern on it, and then also the beautiful, if not functional, abalone bag. Next up, we have Grace, who is wearing a bio-ball top and an agar knit skirt. This agar knit skirt flew from Chicago to Sacramento on a yoga ball as a checked bag to make it to the show intact, and has some very cool structure to it because of that. Up next, we have Maddie. So we had to put the plastic dress on the Urban Evolution person. So we were able to find some bubble wrap and discarded plastic from around the lab and piece them together to make this beautiful garment. And then, of course, plastic dress was littering. But we have Claire here, the least functional, most audacious waiters we've ever seen. She is wearing the fish leather glove, which I think also made its way around. And has a fun smell to it as well. And she was carrying a very pretty bag too, so I wanted to highlight that. So yeah, that's kind of the fashion show. There's going to be a final walkthrough in just a moment, but it was really fun to get to do this as a part of my dissertation and put on this event. And as you saw from those 9 CPB affiliate models, it really was a community effort. Yeah, I'll be quiet. Yeah, I don't That's my presentation. I guess I can answer one question if someone has one. Not. Oh, Joe had a question.
I have a question. I was very interested when you led with the wedding dress, as opposed to ending with the wedding dress as
Yeah.
most. I was wondering if you could speak on that.
Yeah. There was a lot. We went back and forth on that decision many times. It came down to wanting to put the waiters last. So in Claire's outfit, there was some scrap material from every other piece woven into the waiters that Claire was wearing. So we wanted to put that at the end because it was kind of putting all the different techniques and materials that we'd use across the show into one piece, and that felt like the finale, even though a wedding dress is a finale itself.
Well done.
Right? It's like, I liked it because